Hello, everyone, and welcome to our PCE3 session, the last one on transitions from prebiotic chemistry to contemporary biochemistry. And in this session, we're going to have 5 speakers. Each of them will have 15 minutes, about 12 minutes per presentation, and then a few questions. But since our last presentation has been withdrawn, we would like to invite all speakers to come up to the stage at the end of the session, and we'll have about 15 minutes for questions. So I didn't say, my name is Moran Frankel-Pinter, and I'm an assistant professor at the Hebrew University of Jerusalem. And with me, I have my co-convener, Dr. Anton Patra from Georgia Tech. He's a research scientist and a co-I in the NASA Center for Origins of Life. So we're very happy to start. And I'll just say that, as I mentioned, we have 5 presentations, one of which, the second one, is going to be recorded. But other than that, all of them are in person. And the first speaker is actually not going to be Cesar nor Sylvain, but it's going to be Bradley Berker. So we have a surprise. Bradley Berker, who is a NASA postdoctoral management fellow at NASA headquarters. So Bradley, come in. Hello. Great.
Thank you, Moran. Yes, Cesar is really sad that he couldn't make it to AbSciCon this time. It's always one of his favorite conferences to go to, but international travel is still a giant headache today, so he got delayed at the last moment. So I'm pinch hitting for him on this work that we work together on. So I'll be talking about the shared prebiotic origins of pterins and purine nucleosides in a warm little pond model. Now, I just find it helpful to start out with what environment we're talking about so we can all get on the same page when we're talking prebiotic chemistry, as there are a lot of different environments that we like to speculate where life could have come from and where the chemistry could have happened. And so this work, and my preferred model, is the warm little pond. So this involves having some land masses out there with some water on it. And these ponds would be enriched in all sorts of organics. And hopefully you've heard a lot of talks throughout this conference talking about this model and the different organics that are in there. So I'm not going to list them all because it's a quite extensive list, but they're overall highly or fairly reduced organic compounds. And of note that I'd like to point out as key to the work that Cesar and I do is that it's rich in urea and ammonium formate, which would have been likely quite ubiquitous on a prebiotic Earth. And my focus in prebiotic chemistry, what always captures my imagination, is how to form informational carrying polymers, and mostly how to stitch them together. But another important aspect that my research is focused on is how to form the informational units, what we like to think of as the purines and the pyrimidines from modern DNA and RNA. It's likely that early organisms would have used a similar suite of molecules, maybe not those exact ones, but something similar that uses hydrogen bonding to store and carry the information. And so there's been a lot of talks and a lot of research on how to create the purines and the pyrimidines. But one of the things that we notice coming from the synthetic pathways is they not only lead to these specific informational units that we're looking for, but they give a nice diversity of chemistry that isn't often talked about or really explored in prebiotic chemistry. And so doing a lot of the reactions, we notice that these pteridines and pterins also pop out quite strongly from a lot of the prebiotic reactions that we are doing. And so you can see these are quite similar to the purines that we have out there. And in fact, there is a very close synthetic pathway to lead to the formation of either purines or the pteridines that we're exploring here. And so this is a classic TROB synthetic route that starts with the 4,5-diaminopyrimidine, which is core to a lot of the synthetic chemistry out there for prebiotic molecules. And you simply react it via this upper pathway and you form purines. So you react it with formic acid to form the purines. However, under the same exact conditions, instead of having formic acid, if you had glyoxyl down there, instead you form pteridines. And so this branches off distinctly to form these two very similar molecules. And if we go back and we look at the Berker synthesis published in Science in 2019, his group explored how to create purines and pyrimidines at the same time. And this is a pretty great paper, and I suggest checking it out if you haven't seen it yet, as it shows the simultaneous synthesis of both of the informational carrying molecules that are important to us in prebiotic chemistry. But one thing to note specifically down here is on the purine synthetic pathway, this reaction scheme here passes through a diaminopyrimidine intermediate, which could yield pteridines. And so after speaking with Berker, Cesar said that this follows pretty conventional organic chemistry routes, where it's pretty clean, you isolate the compounds, and you can see them come forward. And one of the things that we are always interested in is what happens if you dirty it up. And so we took the initial experiments, and we started with the Berker synthetic pathway, but we dirtied it up by including urea, ammonium formate, and ribose at different steps of the pathway. So we are not isolating anything in the middle. We are just doing one-pot reactions and not cleaning it up any step of the way. And when we did this, we just wanted to start out to verify if this synthetic pathway worked when it was dirty. And what we saw was pretty gratifying. So the reactions in these dirty pathways did yield some of the nucleosides that we were looking for, but it was just a minor product. When we started with the hydroxyl group up here. The major product was a formulated triamino pyrimidine there. However, when we used it with the amine group up there, we did get quantitative yield. So it was nice seeing that the reactions that Berker proposed did work under these dirty settings with varying yields. However, it got really quite interesting for us once we started including ribose and ribofuranose into the reaction. So now we start with the triamino, the triamine, and we start with urea and the ammonium formate from the beginning. Then we go through the synthetic pathway where we did just one acid cycle, and then we added the ribofuranose under a high pH and did some dry wet cycling conditions at 65 degrees. And suddenly we saw a lot of different molecules pop out here. Most notably, the predominant ones were the pterins, or the formulated products. And we got very little guanosine, inosine, and other of the canonical purines from the synthesis. So we started to see this other really important molecule to modern biochemistry start to pop out just from these dirtied up synthetic pathways that we've known and loved for quite a while. And in fact, when we change the ribose concentration, we start to see a really remarkable preference towards the neoterins overall. So when we have a extremely high ribose concentration of 10 to 1 equivalents, we do get the guanine and the neoterins that are still popping out of solution, with the neoterins being the predominant species. But once we lower the ribose to a 2 to 1 equivalent, we don't see guanosine and guanines anymore. Instead, we only see the terins pop out of the solution. So now we have these parallel pathways depending on the amount of ribose that we have. So if you follow the top pathway, where you start with this compound right here, and you add ribose and you take it through 2 different types of conditions with 10 equivalents of ribose, you can get either the guanosine or the terins. However, if you only have the small amount of ribose in there, under either the standard conditions or the urea ammonium formate conditions, then you get all of these neoterins that pop out of solution. And in fact, it gets remarkable. So we wanted to take it one step further and ask, well, what if you don't add the ribose after you've synthesized it? What if you just start with one pot with it all mixed in there? And so we have the urea and the ribose together with our starting material, and there is ammonium formate in here. I just didn't include it in this scheme. And you take it through the acid cycle and then do the dry-wet cycling at the high pH, or you just skip the acid cycling and you go straight to the high pH cycling. Then what we see is quantitative formation of the terins and the neoterins in the solution, and we don't see any guanosine or guanine at all. And so for the last step, since we are running this in a urea-rich solution, we wondered, well, since we've got urea, can we phosphorylate this on some pretty conventional prebiotic chemistry? Because if you have urea present in phosphate, it can form this metaphosphate intermediate and then create organophosphate compounds. So we took this reaction mixture, actually we took the neoterin at the end after we had synthesized it, and then we added the phosphate to the mixture and took it through dry wet cycling at 65 degrees. And we saw a whole bevy of phosphorylated species and dimers and trimers pop out and all sorts of different phosphates adorned to it. So it's a great pathway to making organophosphate terins and organophosphate nucleosides and nucleotides as well. So we have this scheme that you can see in our paper where it can take you through step by step, adding ribose at various steps, but you can start with the prebiotic milieu, take it through some very nice, simple environmental cycling, and end up with, at the very end, nucleotides and these phosphorylated terins. And I'd like to acknowledge our funding sources through the NSF and NASA, through a grant for the Center for Chemical Evolution at Georgia Tech. And my research has also been supported by the NASA postdoctoral program, which was administered by the University Space Research Association at the time. And I'll remind you that you can look for this paper in the next issue of the Chemistry, a European journal, where we've had the honor of making a cover for the issue as well. So thank you for your time.
Thank you, Bradley. We have time for one quick question, if anyone has anything.
All right.
Also, I can ask something really quickly. So in these experiments you've shown, the kind of the endpoint of the experiment, did you also look at what happens throughout the reactions? So to see
So monitoring them.
monitoring the kinetics or product distribution?
Oh, yeah. So, yeah, we monitor each stage of the way, and we can see the different formulated products or the carbamylated products that go along. And so we can see the very interesting chemistry that happens step by step through the process, since we did isolate them and analyze them via NMR and mass spec for each of them. And so we do see that. We didn't do the kinetics nor the thermodynamics for it. So we don't have that, but we do have yields in the intermediates as well.
Okay, thanks, Bradley. So let's thank Bradley again.
Thank you.
So next up, we're gonna have a pre-recorded talk from Liam Longo from ELSI.
Hello, I'm Liam Longo, and today I'm going to talk about Thioester biochemistry in metabolic evolution. Just this April, I started an independent position at ELSI at the Tokyo Institute of Technology, but what I'm going to talk about today is conversations I've had with Sean McGlynn, my former postdoctoral advisor, and my two collaborators, Josh Goldford and Harrison Smith. We're all very interested in the evolution of metabolism, and today's talk is going to contain some of the ideas we've run across as we've tried to come to terms with this really big question. So what is a thioester? The structure of a thioester looks very similar to that of an ester or an amide, except that the oxygen of the ester or the nitrogen of the amide has been replaced with a sulfur. In the hierarchy of transfer potentials, thioesters are considered to be similarly stable or more stable than acyl phosphates, but less stable than either an ester or an amide. And so in biology, we often see that thioesters ultimately funnel into esters or amides. On the right-hand side, I have the Dedew formulation of the thioester world. According to Dedew, primordial amino acids could be activated by reaction with a thiol to generate a thioester derivative of an amino acid. And these thioester derived amino acids could then react with each other to form multimers, or short statistical peptides. In this way, thioesters could have been one of the first systems that allowed polymerization of amino acids. Ultimately, the thioester derived amino acid would be converted into an adenylate, and this adenylate would then go to react to form a peptide. The conversion reaction would involve reaction of an AMP with the thioester derivative to form an acyl phosphate. This reaction is pretty interesting, in part because it echoes some of the results from Racker. And what Racker observed was that the catalytic mechanism of GAPDH involves the formation of a thioester, an acyl enzyme intermediate, that is eventually resolved by attack from orthophosphate to result in an acyl phosphate. Racker interpreted that catalytic mechanism as being an indication that maybe thioesters predated acyl phosphates, very similar to this framing in Dedew's thioester world. Ultimately, these adenylated amino acids would be formed directly by amino-acyl tRNA synthetases, and then those adenylated amino acids would funnel into the translation system that we know and love today. Invoking a thioester world can solve a problem, namely the poor solubility and potentially poor availability of phosphate to early metabolic networks. The question of whether you can have metabolism in the absence of phosphate was addressed in Josh's 2017 Cell paper. He showed that in the absence of any coupling to phosphoanhydride hydrolysis or any utilization of phosphate at all, you can still achieve a network of some 300 metabolites with just the energetics associated with coupling to a thioester. This is one of the first hints that a phosphate-free metabolic core might persist in contemporary metabolic structures. As a naive structural biologist, I wondered how distributed are thioesters and acyl phosphates, another potential primordial energy currency, across the metabolic map of life. So, using KEGG, we identified all of the relevant compounds and their reactions. As you can see, thioesters are more common than acyl phosphates, and so are their reactions. Furthermore, if we look at the KEGG metabolic map, we see that thioesters tend to be more connected to one another. Their reactions are more associated with each other. Acyl phosphates, on the other hand, are more fragmented across the metabolic map. And while it's not shown here, acyl phosphates come in basically two flavors. Either adenolates, where you have an AMP moiety, or where the moiety is just an orthophosphate. Thioesters, on the other hand, are significantly more diverse. Not only are they enzyme intermediates, a feature of thioesters that we can't capture in this analysis, but they're also associated with 6 different sulfur donors that are not part of the pantethine family of compounds. And of course, the pantethine family of compounds would be coenzyme A, ACP, and the like. Returning to my roots in structural biology, I wanted to look at the diverse folds that support thioester reactions. And so what we did is we took the thioester-associated reactions in KEGG and using HMM profiles from a database of different protein evolutionary lineages, we were able to determine what fraction of the protein universe is associated with thioester chemistry. As you can see here, the number of protein evolutionary lineages that are associated with thioesters greatly exceeds the number of lineages that are associated with acyl phosphates. Looking at just the thioester-associated evolutionary lineages, I determined what fraction of the protein families are associated with thioester activity. In doing so, I was able to estimate which families emerged to utilize thioesters versus those families that acquired thioester-associated activities later in their evolutionary history. What excited me is one of the protein folds that popped out of this analysis was called NAT-IV. And so NAT-IV is a quintessential thioester-associated protein fold because the majority of its protein families are associated with thioester utilization. This protein family is fully distributed across the tree of life in archaea, in bacteria, and it is associated with the binding of the cofactor CoA. This CoA binding site is remarkably similar to the binding sites of other nucleotide-containing cofactors, such as the Rossmann binding of NAD and the P-loop NTPases binding mode to ATP. Furthermore, if you look at the binding mode in greater detail, you'll see that while the pantethine moiety and the pyrophosphate moiety are bound very well by this fold, you find that the phosphate and the nucleoside binding tends to be poorly supported. And so what this suggests is that maybe this protein fold emerged prior to the complete development of the CoA cofactor. And so this is kind of an interesting avenue to think about how the coevolution of cofactors in proteins may have occurred. And now let's turn for a second to the limitations of thioesters. And the first limitation that we noticed is that compared to phosphoanhydrides or even acyl phosphates, thioesters cannot activate carboxylic acids. And so here we have a carboxylic acid attacking a phosphorus, and this is going to form an acyl phosphate. This is the reaction that happens essentially in all ligases. The acyl phosphate is then attacked by another compound, completing the ligase reaction. In the case of thioesters, however, if a carboxylic acid were to attack the carbonyl, the resulting compound would be an acid anhydride. And acid anhydrides are extremely unstable in water. And so thioesters don't provide a useful avenue to activate a second compound. Related to this is that acyl phosphate has 2 potential points of attack you could attack either the phosphorus or you could attack the carbonyl, thioesters effectively have only one place that can be attacked by a nucleophile. That means that phosphoanhydrides and acyl phosphates have a lot more chemical flexibility than a thioester might have, particularly with respect to the activation of carboxylic acids. So, does this mean that phosphates are naturally the energy currency in a world where activation of carboxylic acids is important? Not necessarily. Here I have another paper from Josh, this time from 2019, and what he showed is that you can take a metabolic pathway, such as the reverse Tca cycle, and you can replace the steps that were associated with phosphate and use thioesters instead. And so to do this, in this case, he proposed just two additional types of reactions. In the first one, he proposed a primitive mallyl-CoA lyase, and this is a reaction where an acetyl thioester and glyoxylate react to form a mallyl thioester. And in the second reaction, it's simply a trans-thioesterification between a mallyl thioester and succinate. And so with just these two simple modifications, a pathway that would seem to be dependent on the utilization of phosphate can become phosphate-free, and you can potentially circumvent this problem of activation of carboxylates. The second limitation of thioesters that Sean and I have thought about or wondered about at great length is whether or not the hydrolysis of thioesters is coupled to significant conformational change. So we know that ATP and GTP hydrolysis can be coupled to large conformational changes, but actually very little is known about the extent to which this occurs with thioesters. It's hard to show an absence of knowledge, so I have here kind of a cute PubMed search analysis. If you search for conformational change in hydrolysis with no third term, you get about 1,200 hits. If you add ATP as a required third hit, you drop that down to about a half of the total number of hits. But if you use CoA or thioester, you get virtually no significant hits. And so we've wondered whether or not the lack of thioester hydrolysis coupled to large conformational changes is a representation of our ignorance, or if it's perhaps some fundamental limitation of thioesters just due to their size or their hydrolysis properties, which of course would be very different than a phosphoanhydride. If anyone in the audience knows of a large protein conformational change coupled with thioester hydrolysis, please email Sean and I immediately. We would be very curious to hear about it. And with that, I would like to thank you for listening, and I would like to thank my collaborators for our fun discussions over the past few months.
Okay, so as unfortunately Liam was not able to join us online, we are going to move to our next speaker. Let's see. So next up we have Josh Ariola from UCSD, from Uli Müller's group. He will talk about Gdp synthesis by a catalytic RNA.
Thank you, Moran. So today I'm excited to share with you a project that our lab was working on to identify a ribozyme capable of synthesizing the nucleoside triphosphate GTP. So we know that in extant biology, NTPs are involved in many different biochemical processes. Besides being the monomers of RNA, they're also involved in energy, signaling, and certain nucleotide derivatives can act as coenzymes. And the way that modern biology makes NTPs is through the use of protein kinases. What we wanted to investigate was whether or not this catalysis prior to coded protein catalysts could have been mediated by ribozymes. And if RNA is indeed capable of this type of catalysis, then that might indicate an important role for RNA in the replication of an RNA world organism. The replication of which might rely on three important chemical steps. So first we have the synthesis of nucleosides, their chemical activation, and then the polymerization into RNA strands. And our lab was primarily interested in identifying RNA that can catalyze this step in the middle. And the way we do that is we use a technique called in vitro selections. The general principle of an in vitro selection is you basically start with an enormous pool of randomized RNA molecules, usually on the order of 10 to the 13th to 10 to the 16th unique sequences, and you subject this pool to some sort of functional screen. You can also do reverse transcription and PCR amplification and transcription again in order to regenerate the RNA pool. And you can do multiple rounds of selection. And hopefully, if you've set up your selection system well, your pool will then be dominated by active sequences. And the specific activity that we were interested in was the ability of an RNA molecule to bind cyclic trimetaphosphate and thiomodified guanosine and react them together in order to form 6-thiO-GTP. We used a second ribozyme called the polymerase ribozyme as a sort of detection system, and it would take freshly generated GTP and tag it onto the end of the active pool sequence. And we could use this thiomodification as a sort of handle to isolate those active sequences. So there was one major hurdle that we needed to overcome in this selection. So if we were to do the selection in bulk solution, you can imagine a situation in which you have an inactive pool sequence, and because this molecule can freely diffuse, it might get tagged by the polymerase ribozyme such that you're pulling down inactive sequences. And the way that we got around this was to do the selection in emulsion so that each pool molecule was sequestered in their own droplet, and inactive sequences would not be able to form GTP, 6-thiO-GTP, and they would not get tagged by the polymerase ribozyme. One other important thing that I want to point out here is this selection system relied on the coupling of the activities of two ribozymes through their common metabolite of 6-thiO-GTP. So a previous grad student in our lab, Arvin, performed the selection in emulsion, and he made 18 rounds of selection and observed an increase in pool activity starting at round 12. So you might be wondering what this drop-off in activity at round 15 is. He did one round of mutagenic PCR in order to sample the local sequence space of the pool. He also did high-throughput sequencing of each round of selection, and the plot on the right is showing the last 9 rounds of selection, and 5 of the major clusters he observed. A cluster is just sequences that are closely related to each other. So each of these clusters is assigned a different color in the plot, and the size of the stacked plot indicates the abundance of sequences within that cluster for a given round of selection. So I next took candidate sequences from each one of these rounds of, from each one of these clusters, and tested them biochemically. So the biochemical assay that I used was very similar to the selection step, except for a couple key differences. So one of them is that we are now challenging the sequences to use guanosine instead of the thiomodified guanosine. And the polymerase ribozyme is now taking generated GTP and adding it to the end of a radiolabeled oligo instead of to the end of the sequence. And because of this radioactivity, we could then monitor the incorporation of G using gel electrophoresis. So if we look at one of these gels, the upper band corresponds to successful incorporation of G, and our first 2 lanes are our negative and positive control. And you could see from the gel and from the quantification that it appeared that sequences only from cluster 1 were able to successfully incorporate G to the radiolabeled oligo. And we next took the sequence that had the highest percent ligation, sequence number 59, and I don't have time to go into it, but I did a number of reaction optimization and sequence optimization to arrive at an optimal sequence, which we renamed GTR1. So that's how I'll be referring to it from here on out. One limitation with this assay is that we're not really directly measuring the formation of GTP. And so to assess whether or not GTR1 was actually making GTP, we used LCMS. And I'd like to thank Dr. Su for help with these experiments. So from the LC trace, you can see that there's a peak around 2 minutes that's only present when you have 9 micromolar GTR1 or 6 micromolar GTP. And the mass spec data reveals a peak around 524 m/z that's only present in the 9 micromolar GTR1 samples or the 6 micromolar GTP samples, and it's absent in the buffer system. And this peak is indicative of the presence of GTP. So from this data, we were reasonably confident that GTR1 was indeed making GTP. And I next characterized the reaction kinetics of GTR1 and observed, using the same assay that I showed before, as a time course, we observed the rate of the catalyzed reaction around 1.9 per hour, and the uncatalyzed reaction was on the order of 10 to the minus fourth. So that means that the rate enhancement was around 17,000. I also measured the turnover number of the reaction by titrating the ribozyme concentration and holding everything else constant. And I used the saturation point of these curves to estimate the concentration of GTP formed, and compared that to the concentration of ribozyme to determine that the turnover number was quite low for the ribozyme. It's only around 1.7, and we'll return to that at the end. So we also wanted to know if the GTP generated by the ribozyme could now be incorporated into a growing RNA polymer. And we turned to the polymerase ribozyme again. And so this time, we are feeding the polymerase ribozyme. In the negative control, we're only feeding it ATP and CTP. In the positive control, we're giving it ATP, CTP, and GTP. And in our sample, we're feeding it ATP and CTP, and then the reaction products of a GTR1 reaction to see if it can extend a primer on a template. And what we observe is if you compare the no GTR1 condition to the plus GTR1 condition, you can see that there's measurably more extension at the +2 position, which corresponds to incorporation of GTP, suggesting that there is indeed successful incorporation of ribozyme-generated GTP into the RNA polymer. And finally, I wanted to examine the secondary structure of GTR1 using shape chemical probing. So basically the way this assay works is high reactivity to the chemical probe indicates flexibility and single-strandedness, and low reactivity indicates rigidity and is more likely to be double-stranded. So by measuring the chemical reactivity at each position of the ribozyme, you can use that information to construct the secondary structure shown on the right. And I'll just point out a few interesting features of the structure. It appears to show this trihelical junction, and there's an interesting region in the loop on the right that appears to be protected, and we believe that that is making tertiary contacts with either this stem here or this loop here. So to summarize, we used a selection in emulsion to identify GTR1, and we coupled the activity of that ribozyme to a polymerase ribozyme through GTP. And we believe that these triphosphorylation ribozymes strengthen the case for a central role of RNA in an early stage of life. The turnover number of the ribozyme was a little low, 1.7, so that's the subject of some ongoing studies, which I'll share a little bit about. We want to try to do a doped selection in order to identify sequences with higher variance. And dope selection simply means that we are starting with a partially mutagenized pool from the parental sequence. And the starting sequence we are going to use is a slightly shorter variant of GTR1, and that's to enable higher sequence coverage of the ribozyme. And I'd finally like to thank some important people who contributed to this work. Of course, my PI, Uli. Arvind, this was his main thesis project, and he spent a lot of time and effort working on the pilot experiment for the emulsion selection, doing the selection itself, and analyzing the sequencing data. I'd also like to thank Dr. Su from the mass spec facility at UCSD, Sophie for help with analyzing the HTS data, and Doug Magdy for help tuning the droplet sizes for the emulsion selection. And I'll take any questions if there are any.
Hey, really cool talk. So one hypothesis, of course, for the low turnover number is that the ribozyme is stoichiometric rather than catalytic. So have you guys done any analysis to see if the ribozyme is itself modified or spent by the reaction with the metaphosphate?
That's a good thought. We haven't tested anything yet. I think to your point, what we believe is happening is when we were doing the selection, we progressively reduced the concentration of TMP and guanosine so that by the end of the selection, they were quite low. And we believe that the ribozyme is just really holding on to the product. So we're hoping that with the dope selection, we'll be able to identify sequences that can help release the product.
Chris Mayer-Bacon, University of Maryland, Baltimore County. A very interesting talk. I'm curious if you've, with the GTR1 sequence, if that's capable of adding other NTPs besides guanosine, or that addition of other NTPs would require slightly different sequences of RNA?
Yeah, I think that's a great question. It's definitely a subject for future studies in our lab. We are definitely interested in identifying other sequences that can use ATP, CTP, UTP. We haven't actually directly tested it using GTR1, but maybe I'll jump in the lab next week and try it.
It would be very interesting. Thank
you. Thank you for the question, Chris.
Name's Bryce from Georgia Tech. Did you ever test all those other clusters that ended up not working, if it was just because they were on specific for the sulfur derivative of guanosine, or was there probably just an issue of leakiness in the selection?
Yeah. One thought that I had about that is the activity of those. So, sorry, let me back up. We also tested the activity of those ribozymes using 6-thioguanosine. I was just showing the guanosine reactions, and some of those other clusters are active for 6-thioguanosine, but not guanosine. So cluster 1 actually had, I believe, the reverse phenotype where it wasn't as active with 6-thioguanosine, but it was pretty active with guanosine.
Thank you.
Okay.
And let's move on to the next speaker, Roy Black from University of Washington, who will provide the details on how dipeptides bind to vehicles composed of prebiotic fatty acids and are compatible with the vehicle stability and growth.
Get the pointer up.
This is the pointer.
Yeah, but that won't work.
Oh, yes, you need to use mouse.
Should I just use the mouse?
Oh, okay. So
Just use the mouse. Okay, thanks to the organizers for the opportunity to speak. It's late in the last day of the conference, so I'm going to be a bit provocative. Let's see, how do I advance the slides? Over here. If I can get the slides going.
I guess.
Okay. So I'm going to suggest that the big question in the origin of cells is the following. Do generic oligomers, that is with relatively nonspecific sequence, confer a selective advantage to fatty acid vesicles by increasing their stability or growth? A quick reminder of what fatty acid vesicles are. Fatty acids such as decanoic acid are prebiotic amphiphiles that, when placed in water, spontaneously form micelles and bilayered structures called vesicles. So what I'm proposing is that including an oligomer in this system would increase the stability or growth of these structures. And if that's the case, I'm arguing that these effects could have been a key driver of the evolution toward complexity prior to the advent of replication of specific sequences. So we've focused on whether peptides have such effects because peptides are the simplest biological oligomer, resulting simply from the joining of two amino acids. So another way to put it is, does simply joining two amino acids in the context of a fatty acid vesicle have evolutionary consequences? And I'll give you my answer up front. Yes, we found that unmodified, simple unmodified dipeptides increase vesicle stability as measured by resistance to salt-induced flocculation, and increase vesicle growth as monitored by the increase in size upon addition of micelles. And that the dipeptides do this to a greater extent than unjoined amino acids. So why is this important? It means that a population of fatty acid vesicles would change over time, that is, evolve, as those associated with peptides outcompeted others for fatty acids due to their greater stability and faster growth. Okay, I'm going to go over the data in three parts. First, do amino acids and dipeptides bind to fatty acid vesicles? Using diffusion NMR, we've found that all the amino acids that we've tested do bind to somewhat variable extent. Here we're plotting the percent bound to the fatty acid vesicle. And the dimers of these amino acids also bind with somewhat altered relative affinities. Now, one complication with these data is that our NMR signal may be detecting encapsulation as well as binding. But either way, the key point, the key question now is, does such association have any beneficial effects on the vesicles? So let's look at stability first. When decanoic acid vesicles are exposed to a high concentration of salt, they aggregate into these large structures called flocs. And this is bad for the vesicles. They can't grow and divide in this state, so you could say they're dormant. The flocs scatter light to a much greater extent than vesicles do. So we can use the extent of light scattering as a quantitative measure of flocculation. And with this assay, we find that a number of simple, unmodified dipeptides do inhibit salt-induced flocculation, preserving the ability of the vesicles to grow and divide. Now, there is some specificity to this phenomenon. All the effective dipeptides include a leucine residue. And we think part of the explanation for that specificity is the hydrophobicity of leucine. Leucine contains a relatively long hydrophobic side chain, which could actually fit into the hydrophobic core of the bilayer, thereby anchoring the peptide to the membrane. In any event, the key take-home here is that whatever the mechanism, a number of simple unmodified dipeptides do inhibit salt-induced flocculation, and that sets up the critical question, are these effective peptides more effective than their unjoined amino acids? And the answer is yes. Unjoined leucine and unjoined leucine plus glycine do not inhibit salt-induced flocculation. So the amino acids do have to be joined into a dipeptide. So vesicles that acquired and eventually made dipeptides would have an advantage in salty environments. They could continue to grow and divide. So do the dipeptides actually affect the rate of growth? By way of background, if you add a bolus of micelles to fatty acid vesicles, the vesicles will grow by incorporating micellar fatty acids and free fatty acids. So Zoe Todd, with whom we collaborate at the University of Washington, asked whether including a dipeptide in this system would affect the rate of growth upon the addition of micelles. She used FRET analysis to measure the increase in surface area, and from that we can calculate the radius. So here we're plotting the radius as a function of time following the addition of micelles. And you can see clearly that with the addition of the LULU dipeptide, we get a greater rate of growth than in the case with the control vesicles without added dipeptide. In contrast, unjoined leucine does not affect the rate of growth of the vesicles. And Zoe found that other leucine-containing dipeptides also increase the rate of growth. And as we saw when we were looking at effects on salt-induced flocculation, a peptide that does not contain leucine did not affect the rate of growth. So in conclusion, simple unmodified dipeptides increase fatty acid vesicle stability and growth, and they do so to a greater extent than unjoined amino acids. So what are the implications of this? Well, this is what I suggest. As I said, I'd be a bit provocative to try to keep people awake. Evolution began with the simple joining of amino acids in association with fatty acid vesicles. This led to the emergence of a population of vesicles associated with dipeptides because those vesicles could outcompete others for free fatty acids. And I further suggest that vesicles bearing nucleotides as well may have been most favored, but that would have been because nucleotides can actually activate amino acids for peptide bond synthesis, not because the nucleotides polymerized and started replicating from the get-go. And so this scheme suggests that this trinity of membranes, peptides, and nucleotides emerged at the very beginning of the origin of life, and indeed actually drove the progression toward full-fledged cells. And so I'd like to recognize in particular Zoe for all the work on the growth, and Ming-Jen Zhua did all the NMR work. Thank you. I'll take any questions.
Hello, Anthony Bernetti here from Georgia Tech, and I was wondering, you were speaking of testing different dipeptides to see how they worked with these different lipid vesicles. Have you given any thought to selection in the other direction? Could there be particular lipids that would be selected because they would grow better in an environment that had dipeptides?
Yeah, very interesting question. I mean, I think this is a simplified system using just pure one particular fatty acid, and yeah, that would be a good way to go.
Very interesting talk over here. I'm curious about whether or not you have any quantitative information on stoichiometry. So how many of these dipeptides might you need per amphiphile molecule or per surface area of the vesicle to have the stabilizing effect?
Yeah, no, that's a great question, and I've been conferring with Jason Greenwald here at the conference about how you really interpret the NMR data. So it's not easy to really, again, for one thing, to distinguish what's bound from what's encapsulated. So short answer, no, but yes, that would be very interesting.
Okay, so for the sake of keeping up with the schedule, I would like to move on to the next speakers. Let's thank Dr. here, and please stay after this, the last speaker. We will have a 15-minute Q&A session, so you will still have a chance to ask your questions. So our last but not least speakers of this session is Kavita Matanas from Georgia Tech, and she will be talking about molecular memory of chemical systems. Please, Kavita.
Hello, good afternoon, everyone. I am going to be talking about memory and molecules today. Just to get us started, I would like to present a framework within which biochemists see the origin of life. We typically see very small molecules or monomers going to slightly larger groups of monomers and oligomers, which then go to slightly even larger oligomers. And then much later, things get much more complex to give you life. And I guess I really liked what Roy said in terms of provocative statements on a Friday afternoon. So I'm going to go ahead and make one. We in the Williams lab are trying to define this idea of molecular memory at prebiotic chemistry. We believe that the origin and evolution of life is the origin and evolution of sophisticated molecular memory. Now, I believe many people in this community will agree with this statement, but very few people would agree on what constitutes molecular memory. And we are going to try to define it in the next few slides. In order to do that, we are going to ask two main questions. The first one being, what are the strategies that biology uses to record the environmental information? And of these strategies, what are the strategies that can be extended to chemical systems? What are the strategies that chemical systems can use to record environmental information? The presumption being that environmental information was one of the driving factors of evolution, and it's important for systems to respond to the environment to continuously change and evolve. With that premise in mind and with those questions in mind, I'm just going to go ahead and show you our ideas of molecular memory. And we believe that these are the chemical strategies in place in biology today that help biological systems record environmental information. And we're going to go through some of them, and I'm going to talk about why we think so. The first strategy that we believe conferred memory to systems is just one second. All right. The first strategy is, we believe, chemical identity. Now, we have redefined chemical identity to mean the specific structures of molecules used in biochemistry or biology today. Typically, they would be the structure of adenosine, thymine, cytosine, and guanine in terms of DNA. And biology has used these molecules, has used the chemical identity of these molecules in very complicated structures over time, fantastically, to record memory over 4 billion years. And that's amazing. Biology is great at recording memory in these, specifically in these molecules, over a huge period of time. But it extends beyond that. So it extends, chemical identity goes much beyond AC and G and T, and it goes beyond that in terms of DNA modifications, in terms of chromatin modifications. Biology responds to the environment. Epigenetic modifications are the ways in which biology responds to the environment and incorporates changes or alterations into the chemical identity of the genetic code to respond to the environment and adapt or evolve. And this is what we believe is one molecular strategy, also what we call molecular memory, that biology uses to record information. The problem with chemical identity, the problem with tying ourselves to the identity of these molecules, is that they can only take us so far. One of the earlier papers in our lab actually talks about how many of these small molecules, even polymers, are heavily accepted and heavily adapted after the origin of life. They have been they have changed their function, and they're doing very different things from what they did when life started. So it's tricky to understand chemical identity in this space, which is which predates the origin of life. So what else can we learn from, or what else can we look at? And one other strategy that we can look at is conformational changes of biopolymers in biology today. This is not specific to proteins, although in this case I've used the example of protein because phenotypic plasticity and prion-based inheritance is something that has given us a lot of information about the way biology records environmental information. Responses to temperature changes have been fantastically recorded in prions, and actually they transverse generations. They have been prions sometimes last up to 10 to the 5 generations, which was very, very interesting to us when we discovered this idea. And this is independent of the genetic code. This is independent of the identity of these molecules, and this is independent of the structure of the organism itself. So this is found in yeast, in bacteria, in multiple different organisms. But the conformational changes are ways in which molecules can record information and pass it on to generation for variable amounts of time. Although this one is a long-term memory of molecules, of course, we can also account for short-term memory in terms of conformational changes of signaling proteins, conformational changes which will then connect downstream pathways for detecting the environment and recording it for any period of time. So hence, we believe that conformational changes in biopolymers are a form of molecular memory in biology today, and we can definitely learn from them. The other strategy, and in our case, the last one in our list, is chemical gradients. Chemical gradients are a very, very common strategy used in biology, independent of the identity of the molecule. So in here, we have the potassium sodium gradient across a neuron, which is commonly used to respond to environmental cues. May it be in the first ever neuron formed, where that just gave a survival advantage to the organism, or in very complex mammalian brains, where we have information stored every minute to form memories. Chemical gradients can also be, as I said, independent of chemical identity, which can be gradients of nutrients, nutrient gradients, which are taken advantage of by bacteria and by demonstrating tumbling behavior, or they may be small molecule gradients, which bacteria use in a fantastic way for quorum sensing and responding to the environment going into biofilm formation. Independent of the identity, again, chemical gradients, we believe, are a molecular memory which exists in biology today. There are some others which I'm not going to go into too much detail about, but just to conclude, although biological memory itself looks very different, these are some of the underlying chemical mechanisms which we can take into the space that is prebiotic chemistry and study them in that space without the constraints that biology places on memory. And as you see here, although chemical identity and conformational changes might only take us so far back, we can very reliably and very confidently study chemical gradients in molecules. We can study reaction cascades, we can study feedback loops in small molecular system and gain more understanding in them, about them, to learn more about prebiotic chemistry without worrying about whether they might not be completely replicated in biology today, because we know they are. With this, I want to end the first part of my talk, which focuses on the principles, and move on to the experimental aspect, wherein I say we at the Williams Lab obviously have picked one such strategy for molecular memory, which we call your Ensemble Memory. And we are studying this in a system that is chemically evolving. And here I would refer to Dr. Moran's talk, where she spoke about the details of this chemically evolving system. For my talk today, I'm just going to say that we took a bunch of small molecules and wet-dry cycled them for about 15 cycles at 45 degrees and conducted an HPLC analysis on them. For the chemistry, I would again recommend you see the talk or the preprint given below. We found that this group of molecules are capable of changing their chemical landscape over a period of 15 cycles only with the addition of water every 2 days. And this was fantastic for us. We saw a change in we saw a change in the chemical landscape. We saw chemical evolution. We saw open-ended change in a system that required us to do nothing else but add water. And the chemistry behind this was really amazing. And the analysis has been covered in this paper here. But what we extended this into was we used this as a chemically evolving system, and we decided to study memory in this. We decided to study how this system is capable of recording, in our case, environmental perturbation. So if the control, we considered the original system, which changed every cycle, as the control, and we introduced a perturbation. So just at one cycle, at cycle 2, we shocked this system at 65 degrees and then took it back down to its original format and treated it exactly the same for the next set of cycles. And we see that we can actually record this perturbation in the chemical landscape, and the perturbation is very evident in the HPLC spectra, and we can confidently say that the environmental perturbation alters the chemical landscape. And further analysis of the HPLC spectra can tease out some more information about the memory of this ensemble, but we can definitely see how studying ensemble memory in a small system can give us more information and can give us insights into the origin and evolution of molecular memory at prebiotic chemistry. As a conclusion, I would just like to reiterate that biological systems are known to use genetic and non-genetic strategies to record environmental information. Study of chemical systems, the way that chemical systems use non-genetic strategies is definitely going to benefit this field. I would also like to say that I was really happy to see a lot of talks in this space doing that, and I think that's great. With this, I would like to acknowledge the Williams Lab, specifically Dr. Williams, Moran, and Wahab. We've been working together on this project, and of course, the Williams Lab as a whole itself. Thank you.
Let's thank Kavita for presentation. And so we have time for a couple of questions for Kavita. Meanwhile, all the other speakers, please come to the podium. So we will have like 15 minutes Q&A session. If you still have all your questions, there are a few questions online, so we will try to cover all of that. Any questions for Kavita? Yes, please.
Hi, Lynn Rothschild, NASA Ames. Kavita, that was great. As you know, I really am a great believer in what you're getting at. And I think what you're really getting at is a very fundamental property of life that's even bigger than what you think. That chemistry and physics is ahistorical. I don't care where the water molecule I just drank came from, it's water. But if you take 100 people and put them in a room and ask, what do you think about phosphine on Venus, you will get a range of answers. It depends on their history. And I'm not convinced, having heard this twice, that you're not actually getting at that very step of the difference between chemistry and life. And so I'd really encourage you to think even more broadly on it. And by the way, also morphological inheritance. As an old protozoologist, there is literature on inheritance of reversal of kinetics and so on in ciliates. So, keep it up.
Thank you. All right. Absolutely. I will definitely look into that.
Chris Mayer-Bacon, University of Maryland, Baltimore County. Great talk. Your mention of chemical systems and chemical ensembles that you're able to reconstruct environmental conditions from those ensembles. It reminded me a bit of some autocatalytic modeling work out of David Baum's lab. So I'm curious if you're familiar at all with that work, or if that has kind of played into your thinking about chemical ensembles and how they grow and evolve.
Yes. So did you say David Baum?
Yeah.
Sorry, yes. I was here for the talk in the earlier session. I guess Lena, one of his PhD students, gave the talk, and it was really, really interesting to me. I did not know about it before, but going ahead, it would be very beneficial for us to look into that. That's for sure. Absolutely.
Thank you.
Thank you.
All right, let's thank Kavita, and let me once again invite all speakers from this session upstairs here. Bradley. Yes. Or yes, please take a seat. And so you are all welcome to ask questions to any speaker, but we can start with sketching up a few questions from the chat.
Yeah.
The first question we have from Klara Hlouchova from Prague. Thank you, Roy, for the interesting talk. I'm interested in whether you have also tried or thought about trying longer peptides and include other hydrophobic amino acids into your study.
Yeah, testing a longer peptide is the first thing we're going to do when I get back to Seattle. So we don't have the answer yet. And next time I'm going for trimers, yeah. It gets more expensive.
Okay, next one we have also on the chat from Uli Muller. That's also for Roy Black. So D-alanine seemed to help vesicles on an earlier slide better than dileucine. The final analysis was focused on dileucine. Was there a reason for choosing leucine over the likely more abundant alanine amino acid?
Sorry, could you repeat that?
Yeah, so the question was that in the earlier slides that you showed that dialanine seemed to have a better effect, and then you focus on dileucine. So what was the reason? And so dialanine would be more abundant on the prebiotic earth than...
Right. Yeah, no, this started out essentially as a screen. I, bought about 20 or whatever different dipeptides, and dileucine was the one that had the most effect.
All right, so any other questions you've had from the audience here? Yes, please. Go ahead.
Yes. Vladimir Subbotin, University of Wisconsin. I have a question to the same presenter. These dipeptides that appeared inside vesicles, is it flip-flop or it's a free peptide?
Sorry, could you repeat that?
Yes. You mentioned that you have a dipeptide appeared inside vesicles. Is it a flip-flop in membrane flip-flop from outside to inside, or they are free dipeptide?
So how do the dipeptides get in? Is that essentially the question?
Yeah, is it are they still bound to membrane, and it's just flip-flop from outside membrane to inside membrane?
Yeah. No, I assume that they are getting through. Again, if you look at some of Jason Greenwald's work, he's looked at the penetration of a variety of amino acids and peptides, and these are very leaky membranes, right? It's just a decanoic acid membrane. Mm-hmm. So we assume that they are getting inside and, right, could be acting from the inside as well as the outside.
So they can further participate into enhancing durability of vesicles.
Sorry, because I'm still not.
So they can further participate in enhancing durability of vesicles, because if they are bound to inside membrane, they can flip-flop and appear outside.
I assume they can go in and out, and again, we're assuming that the mechanism is the binding, but we haven't even proven that. So.
Thank you.
Okay, please, Tim.
Hello, my name is Tim Sokolsky, University of Wisconsin-Madison. I also have a question for Professor Black. And I was wondering if you have examined the role of the interactions between charged amino acids and vesicles out of these amphiphils, like economic acid or desalamine, which are also charged at different pHs, of course.
So could you repeat that?
Oh, yeah. I was wondering if you examined the interactions between charged amino acids and charged vesicles out of charged amphiphils.
Well, I mean, the decanoic acid vesicles will, this is a neutral pH, bear a negative charge, a partial negative charge. So that's one direction we really want to go in is is there interaction between the amine groups of the peptides or amino acids with the carboxylates of the head groups? That's one possibility. It's a really interesting question. Why would the dipeptides have these effects that the unjoined amino acids don't? The pKa of the amine is different in a peptide as compared to an unjoined amino acid. Whether that's significant enough to explain it, I don't know. You've got the peptide bond itself, which could be interacting. And the other thing I thought about is the amine and their carboxylate are further removed from each other in a dipeptide, allowing a different range of relative positions, and that could be critical. So those are my thoughts about how they could be interacting.
Next question.
Hey, how's it going? This is Sean Brown from the University of Maryland, Baltimore County. And I had a question for Dr. Black. Great talks, everyone. Have you taken a look at possible other amino acids that are hydrophobic and aliphatic, like long aliphatic side chains, besides leucine?
That have hydrophobic? Yeah, actually, very interestingly, isoleucine does not have the effect on the stability, at least at the concentrations we tested. So that suggests that, isoleucine, of course, has the methyl group coming off right at the beginning of the side chain. And so that might block this notion that the side chain actually has to penetrate into the hydrophobic core of the bilayer. So, yeah, that was very striking. That blew me away.
Thank you.
Hi, Jessica Bowman from Georgia Tech. My question is for Bradley. This is the first time I've tried Bradley. First time I've heard of these pterins. I was wondering if you could comment on the prebiotic relevance and their potential for uses in informational polymer.
So you wanted the comment on the pterins in what regards?
If you could clarify their prebiotic relevance and potential for incorporation into an informational polymer.
Well, I don't think the pterins are important necessarily for the informational polymers. So they don't exhibit the same sort of like hydrogen bonding that we see in what we like for modern pyrimidines and purines or protonucleic acids. But what we do see in modern biochemistry, they play a really large part in supportive biochemistry and as cofactors for a lot of important reactions. And so we're really intrigued by how they could have supported the nascent biochemistry during the prebiotic, the prebiotic chemistry of the early Earth as well. And so that's how I look at it there. Yeah, so I don't see any hydrogen bonding necessarily capabilities that would make them competitive with the other options available at the time. So that's what really excites us, because if you're looking in the prebiotic world, it's not just like each polymer or each type of biochemistry operating in isolation. There's a lot of unexplored chemistry with the cooperation between them that we really have to uncover. And so we view this as like one step in helping us to view the different associations that could have happened, to know what could be present, and then the next steps would be to figure out how prebiotic chemistry could utilize them compared to modern biochemistry.
Thank you.
No problem.
Mark?
Hi, I'm Mark Ditzler from the NASA Ames Research Center, and I just had a comment related to Roy's talk. It is interesting that the most hydrophobic dipeptide seems it actually is very similar to Kate Alamalo when she was in Jack Shostak's lab, had shown growth of fatty acid vesicles by taking up mice cells in a system where they were generating phenylalanine leucine dipeptide. And so it's also, it's a very hydrophobic dipeptide. And there's actually a paper that just came out last year from Ames, Andrew Powerilli and Chen Yu Wei did some simulations looking at that peptide and a couple of other hydrophobic dipeptides. And they saw in their simulations that the more hydrophobic it was, the faster the uptake was. And they actually have it's an interesting paper. They have a mechanism where the hydrophobic dipeptides cluster together, form a hydrophobic patch, and that allows a stalk to form between the micelle and the vesicle. So it seems like this is consistent with that, and that likely the dileucine, was it dileucine, is probably doing the same thing, making a hydrophobic patch and supporting uptake of micelles.
Right, yeah, I looked at that, and that may be true, but with that peptide, they had both ends blocked. So it was a very hydrophobic molecule, and with the amine and the carboxylate both blocked, that precluded some interactions that I suspect are going on here too.
Yeah.
Yeah.
And so anyway, but yes, the fact that the simulations there was a clear correlation between hydrophobicity and activity, I suspect, is telling us that even with the non-block, that it's the hydrophobicity.
Yeah, it is consistent.
Hi, my name is Sosan Wahbib from University of Arizona, and I also have a question to Roy Black. It's kind of adding up all the questions that have been asked, but I specifically was wondering the kind of bias towards certain amino acids this kind of mechanism does, and if that bias towards certain types that are better encapsulated or selected for by this mechanism, if it's consistent with the sort of trends we see in amino acid composition of very, very early protein, per se.
Right, yeah, great question. I've looked at that very, very casually and don't see it readily, but the next session we're going to hear more, there's a 3 o'clock session, we're going to hear more about what amino acids were incorporated or utilized early on. No, it doesn't pop out right immediately.
Okay, we're almost done, but there's just one more question for Bradley on the chat. So that's from Uli Mueller. So are the pteridines as photostable as the canonical nucleobases?
That I don't know. I wish that I did. So I did a lot of work on the analytical aspects of it, and Cesar's the one who did a lot of the reaction synthesis and those pathways for that. I know he did UV for part of the reactions early on, but I cannot comment on the relative stability between them. I think that's in our paper, but I'm not 100% sure. So if it's important, I suggest emailing Cesar on that. I wish I could speak to it, so I apologize.
All right, thanks. So let's thank all speakers, and thank you everyone for attending.